See, nature has provided us with sustainable ways to create advanced materials. One such innovation is the biological synthesis of nanoparticles using your microorganisms like bacteria, fungi, and yeast. So, today we will discuss how these organisms act as tiny biofactories for nanoparticles production. So, our today's topic is synthesis of nanoparticles from bacteria, fungi, and yeast. So, as we know that nanoparticles, they are extremely small particles. They range usually between 1 to 100 nanometers. Their biological synthesis is eco-friendly, they are cost-effective and safer than many chemical methods, right? So why we were using bacteria? Because bacteria, they are most extensively studied organisms for nanoparticle synthesis because they grow rapidly, they are easy to culture in laboratories, they can easily be generated, they can be genetically modified, and their enzymes help convert metal ions into nanoparticles. So there were two methods of synthesis from bacteria, which is intracellular and extracellular. Intracellular, which is inside the cell in the cytoplasm or the cytosol, and in your extracellular, it will be the outside the cell on the surface or between the cells inside a colony. So, the first is intracellular synthesis of nanoparticles by bacteria. Here, the first point is bioaccumulation. What is bioaccumulation? It is the gradual buildup of substances like we are having pesticides or heavy metals in living organism over time. Okay, so basically it occurs when any organism absorbs a chemical from its environment or diet faster than it can break it. That is the bioaccumulation. Or in order to release the intracellularly synthesized nanoparticles, additional processing steps such as your ultrasound treatment or reaction with suitable detergents are required. Suppose we were having this flow chart. Firstly, we are having your intracellular biosynthesis. Then, entrapment was done by positively charged metal ions with negatively charged cell walls, followed by the enzymatic bioreduction of metal ions within the cell wall. Then it forms your nanocluster, and nanocluster will form nanoparticles which will diffuse off cell wall. Okay. Then next we are having synthesis of your silver nanoparticles. So basically enzymes and biomolecules, they reduce silver ions into silver nanoparticles. So we are having your culture supernatant of bacteria that can be used for synthesis of your silver nanoparticles. Like we were having uh, E. coli, your Klebsiella pneumonia, your subtilis, ठीक है, your Enterobacter and cloacae, and your Bacillus licheniformis. These were widely used. Also, lactobacillus strains can be used for synthesis of your silver nanoparticles. After silver nanoparticles, we were having synthesis of your gold nanoparticles by bacteria. So we were having your Bacillus subtilis 168. It can reduce your uh, silver ions into your metallic gold. Okay, your silver ions into your metallic gold, producing octahedral nanoparticles, which is ranging from 5 to 25 nanometer in their dimension. After this, your iron nanoparticles, they include your magnetite and greigite nanoparticles, right? इसके बाद the organism which is widely used here is your actinobacteria species. After completing intracellular synthesis of nanoparticles by bacteria, moving forward to the extracellular synthesis of nanoparticles by bacteria. So your extracellular synthesis occurs through biomineralization. Okay, it occurs through your biomineralization, biosorption, complexation, or precipitation. Basically, this method simplifies nanoparticles recovery because particles are formed outside the cell. Okay, so pH changes can influence nanoparticle size and shape. So, with the change in pH of the solution, various shapes and sizes were formed. Then this is the diagram for extracellular synthesis of nanoparticles. Like firstly we are having your extracellular biosynthesis. It will form the production of your extracellular reductase in the cell-free supernatant. Then your enzymatic bioreduction of metal ion within cell-free supernatant, and at the end it will be the formation of nanoparticles within the cell-free supernatant. Then after bacteria we are having synthesis of nanoparticles by your fungi and yeast. So fungi and yeast, they are excellent producers of extracellular enzymes. Basically they grow quickly, they are easy to maintain and efficiently synthesize metal nanoparticles. They are reducing enzymes convert metal ions into nanoparticles outside the cell. Okay, so fungi and yeast, they are very effective secretors of extracellular enzymes, and number of species grow fast, and therefore culturing and keeping them in the laboratory is very simple. Basically they are able to produce metal nanoparticles and nanostructure via reducing enzyme extracellularly. So this is the flowchart of the synthesis process, and for advantages include your eco-friendly production, their low cost, reduced toxicity, they are easy scalability, and they control synthesis. So your bacteria, fungi, and yeast, they act as biological factories for nanoparticle production. So nanoparticle synthesis using microorganisms is an important area of nanobiotechnology or your nanoscience to understand your intracellular and extracellular mechanisms that help develop sustainable methods for producing nanoparticles, which is used in your medicine, agriculture, electronics, and your environmental applications. So with this we have completed our today's topic, which is the synthesis of nanoparticles from bacteria, fungi, and yeast.